Hello, everyone. Thank you for joining us today for our ALS Webinar Wednesday series. My name is Leslie Venegas. I'm the Marketing Manager for ALS North America, and I'll be facilitating the webinar for today. A few helpful notes before we begin. If you're having technical issues with our webinar platform or any questions, type your message or question using the chat function at the bottom of the screen. You can also select the hand icon to notify me of your status so I can assist. All questions regarding the webinar material will be answered at the end of the presentation and can be submitted using the Q&A function in your toolbar at the bottom of the webinar. Our presenter today is Louis Wagner. Louis Wagner received his Bachelor's of Science in Biology and Chemistry from Simon Fraser University in 2003. He joined ALS shortly after his graduation, establishing a new satellite laboratory for ALS in Fort Nelson in 2005. Shortly thereafter, he became acting supervisor of the ALS Calgary Inorganics Department. Louis took on his current role of ALS Canada National Technical Inorganic Specialist in 2008, and as such has led and conducted numerous analytical method development projects in the field of inorganic chemistry, including anions by IC, metals by ICP-MS, automated colorimetric flow systems, and arsenic and selenium speciation by HPLC and ICP-MS. Mr. Wagner is a key member of the ALS Canada National Technical Group, which develops, implements, and promotes innovative best practice techniques, as well as national methods that are used by ALS laboratory locations across Canada, and is widely regarded as one of the top inorganic analytical chemistry experts within the ALS Global Lab Network. Today, Louis will be focusing on selenium speciation in waters in Biota. Louis, thank you for taking the time to deliver this presentation. I now turn it over to you.
Thank you for the amazing introduction. I will try to live up to that. So my name is Louis Wagner, and I didn't hear in that introduction where I am currently located. So I'm located in Vancouver, British Columbia, Canada. So I'm on the West Coast. So good morning to everyone who's on the West Coast, and good afternoon to anyone further east. We always like to start a webinar or a presentation with a little safety moment. And since I'm going to talk about selenium, well, that's going to be the topic of our safety moment. And selenium is an essential element, so you want to vary your food and diet. Lots of foods can have different amounts of selenium. So vary your diet, read the ingredients of any supplements, make sure you get enough selenium in your diet. And an interesting note is that Brazil nuts is one of the highest sources of selenium. So a single nut is your recommended daily dosage. More than a single nut is too much selenium. So your safety moment is try to remember how many Brazil nuts you've eaten from the bowl of mixed nuts, which we start to see around Christmas time. Also, since it is the age of COVID, just a reminder, please be safe, please be kind, follow health authority recommendations. So, brief overview of my presentation. What will I be covering? I'll briefly be covering what is selenium, why selenium is important, selenium regulations, and briefly on the regulations because it is a very evolving topic and constantly changing. Why is selenium speciation important with relation to selenium regulations? Some analytical challenges related to selenium speciation, and then we'll open up for questions. So, what is selenium? Well, selenium is a naturally occurring metalloid, often found in metal sulfide ores, and it exhibits both metallic and non-metallic properties. The properties are often similar to sulfur, tellurium, arsenic. The main anthropogenic sources is typically mining and power generation, oil refining, and agriculture. Waste rock associated with coal and uranium mining, oil refining, and smelting also have a potential to leach selenium if water and oxygen come in contact with the waste rock. So why is selenium important? Well, selenium is an essential micronutrient. It is incorporated into amino acids, proteins, and antioxidant enzymes. It is essential for the health of people and animals and some plants. An interesting note is a lot of the soils in British Columbia are deficient in selenium, so it's often added in fertilizers. Deficiencies can correlate with things like hyperthyroidism, heart disease, cancer. And there also have been recent studies that show selenium concentrations have a protective effect on arsenic and mercury poisoning in the environment. However, selenium is interesting in where it has benefits, it also quickly becomes toxic. So there's only an order of magnitude that separates the tolerable level of selenium in humans and toxic effects. So, go back to the Brazil nut. One is good, a handful is toxic. Excess selenium in diet can cause reproductive issues and abnormalities in egg-laying vertebrates. So it's affecting the reproductive health of these animals. I post here a really good document. It's quite large and goes into huge detail on selenium toxicity and its effect on humans and other organisms. So this presentation will be provided to any attendees, and I would recommend, if this is part of what you're interested in, to check out the screening assessment document from Health Canada and ECCC. Selenium regulations. Now, selenium regulations, they're currently constantly evolving. There's three primary avenues that regulations are targeting, and this is water concentrations, so water samples, whole tissue, fish and wildlife. And this is particularly of interest to assess diet-based exposure, particularly, say, First Nations populations in different parts of the country. And also eggs and ovaries, which would assess toxic effects in the population of these animals. So is the selenium affecting the reproduction of salmon in your streams, for example? My first regulation slide here is just showing some total selenium guidelines currently in Canada. We have a National Aquatic Life Guideline of 1 microgram per liter. This is somewhat outdated because our understanding has changed recently. There is a fish tissue regulation at 14.7 parts per million. There's a few others. There's the CNSC has their own guidelines, and provincial guidelines are changing rapidly. Currently, the most default, we have BC and Ontario with 2 different ones, 2 microgram and 100 microgram recently, and another province has adopted British Columbia values. The takeaway here is that guidelines are changing. You need to keep up to date. I would talk to your regulators if this is what you're really worried about, and talk to our client services group, who also may be more up to date than I am on some of these regulations. I don't want to spend too much time on the regulations. It's not really my specialty, but here's another slide that summarizes, including when these regulations came into effect. From freshwater guidelines, there's an alert guideline and a threshold. Dietary guidelines, interim at 4 micrograms per gram. Some sediment, TISH. The TISH egg, ovary, whole body, these are more recent additions. For any US clients or who are interested, there's also American guidelines from US EPA, updated in 2016 for different types of waters. I'm just going to let the slide speak for itself here. There are also a lot of individual state guidelines, and so it can get pretty complicated. Okay, so why is selenium speciation important? So all these regulations from the previous slides are total selenium or dissolved selenium. So why is the speciation important? Well, selenium inorganic forms, they enter the food chain and bioconcentrate. So they get converted from the inorganic forms to some other forms of selenium, for example, selenocysteine and selenomethionine. And recent research indicates that a better understanding of the speciation is needed to understand the impacts. So in order to predict the behavior and impact of selenium in the environment, including how bioaccumulation to toxic levels can occur, the concentrations of selenium species need to be understood. So I have a nice diagram here kind of showing how it cycles through the environment. So selenium in the environment predominantly exists as the dissolved oxyanions, selenite and selenate. As it cycles through the ecosystem, additional compounds become present. So as a general observation, what you might find is a selenium source to a river. Closer to the source, it may all be selenate. Further downstream, partway through, it might be selenite and selenate, or selenite and some other species. And as you get to the ocean, may no longer be any selenate. It may all be selenite or other forms. And that is actually an example of the San Juan River, what they see as they track selenium species down the river. Also, the invertebrates in the river will cycle into different compounds such as volatile organoselenides. Common species of selenium. So we have our primary inorganic forms, selenite and selenate. These are the predominant forms that make up total selenium concentrations in waters usually. We have other inorganic forms, selenocyanate and selenosulfate. And these are most often produced from power processing plants and refining when fuel source originates from fossil fuels and oils. Biological activity, how that affects the selenium species, we will start to see selenocysteine, selenomethionine for vertebrates and fish. Perhaps microorganisms, we will see methylation. So that might be dimethylselenide and dimethyl diselenide. And these are volatile species of selenium. There can be oxidative processes which create methylselenic acid or dimethylselenic oxide when it reacts with some of these biological offshoots. So, and then there's other species as well, elemental selenium. And really, once we start looking at biota and how organisms are processing it, there's hundreds of different species. There's really too many to list. So let's go back to total and dissolved selenium. So the analytical methods for total and dissolved selenium. Typically, this is analyzed by ICP-MS. Total selenium would be perhaps EPA 200.2 or 6020B. And briefly, samples are collected in an HDPE bottle, preserved with nitric acid, and digested with a mixture of nitric and hydrochloric acid, and analyzed by ICP-MS. The difference with dissolved is these samples will be filtered prior to preservation. Our typical analytical methods for selenium speciation. Some of the older school type of methods would be hydride generation or cryogenic hydride coupled to various detectors, either AA or fluorescence. You can also have an IC or an HPLC coupled to an ICP-MS, and the ICP-MS can be any of the types from high res to collision cell to triple quad. Headspace GCMS is also useful for separating and analyzing volatile selenium species. So since I'm a laboratory guy, we're going to talk a little bit about some laboratory challenges. And there's some questions when you want to do speciation of any kind. The first question is, can we separate the species? So this is where the instrumentation comes into play. The HPLC will separate most ionic species. If it's something volatile, then we can use a gas chromatogram to separate the GC. So that's the first question. Can we separate the species from each other? The next question is, well, is it even detectable? So some species can pose difficulties in detection. Volatile species would be an example of that. If we lose it before we even run it, we can't detect it. Elemental selenium also is insoluble and often sticks to containers. So if that is of interest, there's a challenge there. Once we've sorted out, can we separate it? The next question is, can we identify it? So one of the main limiting factors is often the availability of standards. Sometimes I can purchase these standards, and sometimes they might require in-house synthesizing. To identify a peak, so when we do our speciation, I'll show some slides in a little bit about what that looks like. We often will see peaks that we do not know what they are, but we will report them semi-quantitatively based on selenium response. So if we see some unknown peaks, we'll give that information to the client. We can identify that peak if we have a standard and can match it, and our standards don't have to be of the highest quality to simply identify and say, yes, this compound is present in your sample. But the last question is, can we quantify it? So can we give you a concentration with a good enough accuracy that is useful to you? And that gets more complicated as we need to have standards that are certified. The best kind of standards would be certified externally, but they are also quite limited in numbers we can find. So sometimes we need to certify these standards internally ourselves. So I'm talking about separation. So what does that look like? I like showing chromatograms. This is just a typical selenium speciation chromatogram. We have 4 species that are quite clear and separated: selenium methionine, selenium selenite and selenate, and selenocyanate. Nothing too challenging with this sample. I like to show, here is a wastewater, and this is actually pretty clean wastewater. So here we're starting to see there's a little bit of selenocyanate in this sample. There's now methylselenic acid, a different species that I briefly mentioned earlier. There's also a peak that we don't know what it is at this time. So that's question mark. But another wastewater can have even more species. So in this example, there's a very large selenium methionine, very large selenocyanate, inorganic species, as well as at least 3 other unknown species. And these by far are not the worst kind of samples we see, as depending on what the process is, we're testing what the wastewater is. If it's part of a treatment process, things get a lot noisier. So, total selenium doesn't always tell you the whole picture. And sometimes the species, 3 or 4 species, don't tell you the whole picture. So in these cases, we need a lot of communication with the client to see what we can give you and what you're looking for. I believe the most species I've counted in a sample was over 30 clear different selenium species present in a sample. I believe that was in a bioreactor. Not unsurprising that would have a lot of species. I like to show some biota. So Here is an example of a terrestrial vegetation sample that is selenium-enriched wheat. So in this case, the wheat only really has one species, Selenium methionine. But in a aquatic organism, thought I'd just zoom in on Selenium methionine, and here we have a Biota sample that has a lot of different unknown species. So really, I think the takeaway here is that we need to collaborate when we're discussing selenium species. Perhaps chromatograms would give you some information rather than a couple concentrations. Or you're looking for a particular species, or you're looking for something specific. So, but what use does Selenium speciation have? Well, I'm going to give two examples where, two case studies where this has helped clients and helped us understand what is going on in the environment. Now, this is a common thing we often see, is dissolved selenium greater than total selenium. And in this case, this was a wastewater treatment plant. So the influent, everything makes sense with our typical regulatory total selenium, dissolved selenium analysis. Total selenium, 50 micrograms per liter, dissolved 45. If we looked at the species, 45. However, in the effluent, something didn't make sense. The total selenium was 20 micrograms per liter. However, the dissolved was much higher than the total. And as well, the species agreed to the total, not the dissolved. So what we did here is we investigated quite a number of years ago. This is not new for us, but so we investigated literature, we did some internal studies, and we discovered that volatile species were likely the cause of this elevated dissolved selenium. So we tested this. So volatile species enhance the signal on the dissolved analysis. So what we thought is, well, let's digest the dissolved. So in this case, it's a filtered sample digested. No difference in the influent, but in the effluent, now the undigested selenium was 100 micrograms per liter. If we digested it, it came down to 15. So that proved that the digestion was critical here. Now, were we losing the volatiles or were we converting the volatiles? So we performed a study to see what was happening to volatile selenium species in our standard digestion. So we spiked a volatile species and ran it a few different ways. We also wanted to test if perhaps preserving in the field might convert these volatile species into an inorganic form. So first test would be no preservation, no digestion, spike it with a volatile species, and we got 100% recovery. So we see this enhancement from volatile species. If we digest the sample, the second row, now we see 79% recovery. So now we're getting recovery we would expect for selenium. So we've converted the volatile species. Now we try just preservation, either at 0 hours, so immediately analyze after preservation and sitting for 24 hours. And in both those cases, we still saw the enhancement. So nitric acid preservation alone did not do anything to the volatile species. And then preserved and digested, which agreed with the previous digested. These results are within our analytical data quality objectives here. So what this showed is our digestion converts the volatile species. We weren't just driving it away. We can spike with volatile species and get good recovery. So this is really important when we see samples that we suspect might have volatile species. We digest the dissolves. Now the client's getting results that make more sense to them with comparing the total and dissolved. But how can we really confirm that there's volatile species? Well, we developed a GCMS method to analyze for the two most common volatile species that are in samples, dimethylselenite and dimethyl diselenite. So these same samples where we did the digestion of dissolved to confirm, to get an agreement between the total and dissolved, we also analyzed them for the volatile species themselves to prove that they were there, which we did. So this would be a way to investigate discrepancies between total and dissolved selenium. We can analyze for volatile species to confirm if that is the reason. And then moving forward, we would digest the dissolved. Another challenge, and now this is a challenge from selenium samples, typically from oil refinery wastewater, where we see, in this case, we have a total selenium of 20 micrograms per liter, a dissolved selenium of 19 micrograms per liter. However, when we did speciation analysis, the sum of the observed species was far greater than we were getting for total or dissolved selenium. And the most prominent species present was selenoscyanate. Now, the issue with selenoscyanate is if it's acidic, it decomposes to elemental selenium. And in this case, this is what is happening with these samples. The client total selenium and dissolved selenium results were not balancing out with their process. They felt they were missing, that we were missing selenium. So we do the selenium speciation analysis. We see that there's selenoscyanate. And the reason that's missing from the total or dissolved is when it's preserved in the field, it turns to elemental selenium, and then that sticks to the plastic of the bottle. And it's very difficult to get off the plastic. So in this case, we could develop some better sampling that will let us analyze for the elemental selenium and include it in the total and dissolved. So this is kind of a special case where the sort of regulatory approach to how you sample for total metals, we're missing some of the metals because it's sticking to the plastic and not coming off. Normally, when you're doing total metals, the preservation is there to help keep the metals off the container. But in this one case with elemental selenium, it's quite sticky. So one approach, we would discuss this with clients and we can come up with an approach to deal with the elemental selenium. Here is a summary of our current species offerings. This is also somewhat evolving as requests come in. Can we analyze for this other species, or maybe can we identify some species in a sample? But generally speaking, this is our more routine offerings. So we offer some Biota species, the inorganic species, selenomethionine, and we also give a rough result of all the other unknown peaks that we observe. In water samples, we offer the inorganic species, selenoscyanate, selenomethionine, methylselenic acid, dimethylselene oxide, and as well any unknowns. And as I mentioned before, we also can offer volatile selenium species. So to recap, HPLC, ICP-MS, and Headspace GCMS can detect hundreds of selenium compounds. This speciation data can provide information that can help scientists understand the dynamics of selenium compounds and make some decisions. And HPLC, ICP-MS is a sensitive and reliable technique for analyzing selenium species and making sense of total and dissolved metals data from complex samples. And that is my presentation. Thank you very much for attending. And now I turn to questions.
Thank you, Louis. Yes, we'll now open the forum for questions. Please type your questions using the Q&A function in your toolbar at the bottom of this webinar. I'll give everyone just a couple moments to type in their questions as I review some brief notes. So we did want to mention the webinar presentation has been recorded. All participants will receive a follow-up email once the recording is available to view. We also post our webinars on our ALS Global website and our ALS YouTube channel under the webinar playlist. I encourage you all to take a look at our wide range of video resources and past Webinar Wednesday presentations. So it looks like we do have one question. Louis, can you please review the main sources of selenium that are introduced into the environment?
Sure. I can probably do this from memory. So the main sources, what we see the main sources would be mining, coal plants, agricultural runoff. These are the main sources of selenium being introduced into the environment. Yeah. I had a slide for it. Does that answer the question? The main source that we deal with here is mining. We see more mining than petrochemical sources, but that just might be the nature of the work we're receiving, particularly from tailings rock piles where this rock has been crushed up and now there's rock containing selenium exposed to water, exposed to oxygen, and that's usually quite a big source.
Okay, thank you. It looks like we have another question. For the chromatographs, how do you know the unknown peaks are selenium species and not some other metal or contaminant?
That's because we're using an ICP-MS, so it's element specific. So all the peaks are selenium. They're not from any other metal or any other compound. So that is an advantage of the ICP-MS side of the instrument. I know with very large certainty that these are all selenium. If we had some compounds that might contain selenium and a sulfur, I can actually confirm it by looking and seeing if sulfur is also in that exact peak. But yeah, it's specific to selenium.
Okay, next question. Which species are related to anthropogenic activities?
Well, inorganic is the sort. That coming out of the like waste rock or the mining, it's in inorganic forms. It's being converted by the ecosystem, by the algae, by the invertebrates, by the bacteria into other forms. And then those forms bioaccumulate.
Okay, next question. What methods is ALS using to differentiate selenite versus selenate versus organoselenium in Biota samples?
That we're using the HPLC ICP-MS. So that's the method.
Okay. In relation to chronic toxicity in fish, what is the best method of testing?
Chronic tox, that one, I'm not sure. That question I would have to think about a little more.
Okay, we can make that response available to the group in an email follow-up. And then the next question is, which species are more toxic?
That one I'll have to look up and reply to. I believe. Okay, no, I'm not going to answer that until I look it up.
No problem. How much the bioaccumulation of selenium be increased by discarding cyanide, discarded into the environment from analytical tests?
Sorry, what was the question again?
It says how much bioaccumulation of selenium is increased by discarding cyanide, discarded into the environment from analytical tests. We may have to follow up on that one.
Yeah.
Okay, and then given the potential implications on compliance, how are the regulators learning about the complexities of selenium speciation?
That one I'll have to follow up on. I know we do a lot of collaborations with both, at least locally, with the regulator and the client, in particular when there are these situations. But yeah, I'm not sure I can talk about that, but we can follow up maybe after.
Okay, in mine wastewater, other metals may be very high, such as iron. Have you had problems with interference, I believe, of other metals, making it challenging to quantify selenium species? Interference.
Not on the species. The system separates out all the species. It's pretty robust. Like we can run from seawaters to clean waters. Biotas are the most complicated of matrices. I would say that there's a possibility that on the sampling side, there may be challenges with things like a lot of iron in an anoxic environment. And this would be something we would probably want to know ahead of time with the client, and maybe we can come up with a recommendation. Things like pour waters or taking water out of peepers, I think they're called. So if it's very anoxic, you might start to get precipitation before you've even put it in a bottle.
Okay, and how do you handle polyatomic interferences with the ICP-MS?
We're using a collision cell ICP-MS, so it is using a reaction gas. So when we're analyzing selenium, it's removing any polyatomics.
Okay, and our final question, I think I saw an amount of tissue required is 10 grams for analysis. Is this the optimal amount? What is the absolute minimum that you might be able to work with?
We can probably work with, yeah, that's an optimal amount to get a good homogenization, but we probably get, we do have methods for total metals where we can go down to biopsy amounts. We could probably do something similar for speciation, but we might have to adjust some detection limits.
Okay, and then we do have one more. What kind of ores are mostly considered as sources of selenium to the environment?
I am not the expert on this, but what I have seen is coal is a very large source, but also some copper mines have a selenium problem. But yeah, I'm not the expert on all the different ores and which ones would have selenium.
Okay, thank you. It looks like we've covered all of our questions. Louis, is there anything else you wanted to add before we conclude the webinar?
No, I think that's everything. And if anyone has any questions, I guess my email is on the last page here. They can contact me or contact your local ALS location, and they can track me down.
Great, thank you so much, and thank you everyone for attending today. Please follow our ALS Life Sciences company page on LinkedIn. That's where we post announcements and registration links to future webinars and other valuable resources and updates. If we have any more questions that come through to the Q&A section, we'll be sure to get those answered, as well as those that are going to be researched offline, and we'll send that to the group as well. Thanks again, and enjoy the rest